you guys. People did pretty well in the lab test. I was glad to see that. I sent out the results last night. So that's a good movement in the right direction. Many of you guys are starting to make some adjustments to how you prepare for this course. And if you'll continue to do that, lecture exam 2 is usually easier than lecture exam 1. Why? Well, 1, because the topics are so similar, and 2, because the topics from lecture exam 1 build on each other to help you understand lecture exam 2. As you know, lecture exam 2 is not this Friday. Lecture exam 2 is going to be not this coming Friday, but the Friday after that. It will be October 9th. So today's September 28th, Monday. This, you guys can see my black pointer, right? Can everybody see the syllabus and the black pointer? Okay. Lecture exam 2 is moved to this Friday here. At this point, there's a good chance you're getting this Friday off. I'll make a definitive decision on Wednesday. But this month, today, we're going to be doing a lab on algae. On Wednesday, we'll do a lab on protozoa. And I'm leaning towards giving you Friday off. Lecture exam 2 is the following Friday. So this week, you guys, we're going to be doing the lab stuff. You want to be in the Zoom meetings and taking good notes and paying attention, but you need to get serious about chapter 24, speciation, and start in on 25, the history of life. I have like 2 more things to do in chapter 26, but you'll see that 26, you guys don't have to do the whole chapter. I have 2 videos to put in there. I'll get that done sometime soon. So the content for chapter 24 is all done. Content for 25 is all done. I got a little left to do on 26, not much. But here's a cautionary note. Notice how 25 is the history of life, and then I have 25 as the origin of life. The origin of life material in chapter 25 will be on lecture exam 3. That's why if you go down here and it lists these chapters, lecture exam 3 has portions of 25. It has the origin of life information on there. So you'll see that when you read in 25, and you'll notice I've told you which pages you can skip for lecture exam 2 and which pages you need to focus on for lecture exam 3. So that's, if you look down at lecture exam 3, if you say, why are we doing 25 and 26? Well, we're doing a little bit of 25, which deals with the origin of life on lecture exam 3. And at the end of chapter 26 is a few pages that is on exam 3 as well. But exam 3 is primarily all of 27 and a lot of 28. Exam 3 is primarily the prokaryotes. It's a little bit of origin of life. Well, I mean, there's going to be a lot of questions. It's a lot of, a number of questions about origin of life, a lot of prokaryotes, and a lot of protistans, and just a little bit from 26. But that's lecture exam 3. And right now, you guys don't care about that. You care about lecture exam 2. Questions about, everybody understand lecture exam 2 is not this Friday, it's October 9th. The Friday after that.
Thank you for clarifying that.
Yeah, remember, if you're ever not sure when the assignment, because we modified the assignments, you go right here. When are assignments due? We're not modifying this one. We'll stick with this. These assignments aren't changing. These due dates aren't changing. Bingo, right there. All right.
How many
And
Sorry.
was there a question?
Yes.
Okay.
So how many pages did you say you want on the jungle?
Well, I got a lot of classes and a lot of papers. I don't remember. So you need to go to the policy, course policies. Go to course policies.
I remember the introduction was like 2 pages.
I think there's 9 chapters.
Mm-hmm.
Okay, it's right here. See, I would have guessed 2 pages per chapter because I got too many classes and too many papers. So you guys have to learn to go to the syllabus. Well, go to the course policies where it'll tell you. Otherwise, I'm going to tell you 2 pages a chapter. It's technically 1.3 pages per chapter. That's the minimum. So it has to be 12 pages. 1.3 means a page and a third. I get very irritated if people provide me less than the minimum. So my advice is go a page and a half to 2 pages per chapter. And then you won't lose points. But it has to be a minimum of 12 pages. And as I think you guys are learning, please don't make the mistake that students do. And sometimes they'll give me a half a page for one chapter and then like 3 pages for another chapter. Well, you're going to lose points because every chapter has to be a minimum of a page and a third. You follow me? So that you just don't write it. So some students will write, most of their paper over 6 chapters, and the other 3 chapters they didn't do much on. Well, you're going to lose points. So please don't do that. All right. Other questions about the jungle paper? But remember, I've moved the due date for that. That's not due until October 30th. Jungle paper is October 30th. Check when assignments are due for that.
Thank you.
We were discussing, does everybody understand why I sent you that email on plagiarism? Hopefully you guys read that. I've ran into some problems in my evolution classes, and the gist of that is I want people to understand that you can get caught plagiarizing even if your similarity report is 3%. And that's what happened with several students in my evolution course. Their similarity reports were low. Why? Because they went to this website and they copied the information and then they rearranged the words. They rearranged the words well enough that Turnitin thought it was an original document. The trouble is these students had all gone unknowingly, right? I got 3 evolution courses and 70 students. They had unknowingly gone to the same website. So after I'm reading a couple of these papers, I'm like, wow, the concepts are the same. And so that's how I caught them. So I need you guys to, we only had 1 student on the transformation report that was bordering on plagiarism. I made a note of it to them. I didn't, I deducted a few points. It was borderline. But usually when we go to the jungle paper and the shape of life papers where I see problems. And even in this class, usually I will get 2 students who will try to plagiarize on the jungle paper, or it's usually in the shape of life paper. Why? Because that's toward the end of the semester. People are busy and students are looking for quick ways to turn in assignments. Please, let's not do this. So you guys understand the policy. You'll get a 0 on the assignment, and then I have the option. I can, even though I usually try to just give somebody a 0 on the assignment, I've already informed you guys, given you a warning. I can just flunk you for the class. And I don't think students fully understand that if faculty wanted to, they can have this put on their permanent transcript. That's why I wanted you guys to read that letter or that email I sent. You don't want a note in your permanent academic record that you've been caught cheating. that usually doesn't go over well when applying for jobs. Does everybody understand? I'm trying to just help you guys out, not only for my class, but for future classes. Not only is it easy for you guys to cheat and plagiarize, it's also easy for faculty to catch it. It's a two-way street here for the ease of this. So please, it's not worth the risk. Does anybody have any questions about my email and plagiarism? And I hope that everybody understands that if you go and copy paragraphs from a website and you rearrange the words so that Turnitin thinks it's original, that is still cheating. You have not written it in your own words. You've paraphrased it. That is still cheating. That's still plagiarism. Does anybody have questions about this? All right.
Chapter 25, History of Life, and Chapter 25, Origin of Life. Is that the same chapter in the textbook?
It's the same chapter in the textbook. The reason I've separated them is the topic of origin of life is on lecture exam 3.
All right.
So when you get into chapter 25, it's the first few pages, I think. There's a section in there where they discuss the origin of life and biochemical evolution. That is on lecture exam 3. So you can see over here, the speciation is done. Chapter 24, content is ready. Chapter 25 is loaded with content. Don't blow this off. You need to get working on that eventually. Hit the load more button. One good thing is you'll notice there's similarities. You guys have already done the stuff on paedomorphosis, homeotic genes, from fins to hands. So some of this is similar. Some of this is a review. The evolution of the stickleback fish, that's a review. Some of it obviously is not. Most of it is not a review. And then 26, good news is there isn't going to be a whole lot to do. I got a couple more items to add, but maybe 1 or 2 or 3, not a lot. But I clearly make sure you guys read up here because I clearly tell you what pages you have to do and what pages you can skip. Tell you that for 26 and tell you that for 25 as well. All right. So 24, 25, and 26 are lecture exam 2. Not this Friday, but following Friday. Let's keep working hard and making adjustments and get a nice performance on lecture exam 2, and we'll start moving in the right direction here. Questions about things before I start the lab? All right. Let's begin our lab. I'll be honest with you. Your second lab test is significantly tougher than the first. And it's harder than the third. So your most difficult lab exam is exam 2. A lot of slides, a lot of images. What we're going to be doing over the next few weeks is exposing you guys to some of the diversity of life forms that exist out there. So we're trying to get you guys an appreciation of different forms of life. There's tremendous diversity of life on this planet. And over the next, well, really for the remainder of this course, the lab focus will be on different types of living organisms. Today's lab deals with the algae. And and then Wednesday's lab will deal with the protozoans. These are not taxonomic names, so they are not formal scientific names. These are informal terms. Algae and protozoa. And they these are all in what are called protists. Protists, protists. We used to have a kingdom called Protista. But then as we learned more and more about these protists, we realized there's a tremendous diversity of these protists. And that what used to be one kingdom Protista is now something like 25 different kingdoms. Well, I don't want to have to learn the names of 25 different kingdoms, so I doubt that you guys would want to do that. So if you ever see the term kingdom Protista, that is outdated. We no longer use that kingdom name. And the term protists is a general term. It's not a taxonomic term. But you'll hear that a lot, protists. So what are protists? Protists are eukaryotes. Protists are some of the oldest, most diverse group of eukaryotes. Remember, eukaryotes have more complex cellular structure than prokaryotes, right? Prokaryotes, compared to eukaryotes, have simpler cellular structure. Prokaryotes were the first types of cells on the planet. They are still the most abundant living things on the earth. But protists are not composed of prokaryotic cells. They are composed of eukaryotic cells. As you'll learn eventually, eukaryotes evolved from prokaryotes. So when we use the term protists, we are talking about a very old lineage of cells, very diverse group of eukaryotic organisms. They are, for the most part, they're unicellular, microscopic. That means they're so small we have to use a microscope to be able to see them. So for the most part, they are unicellular, very small eukaryotic organisms. So basically, protists are any type of eukaryote that is not a plant, an animal, or a fungus. So we will eventually talk about plant and animal and fungal evolution. But protists are the eukaryotes that are not plants or animals or fungi. What's intriguing, though, is that you will find plant-like, animal-like, and fungal-like cells in this group of protists. So the protists contain plant-like, animal-like, and fungal-like organisms. You might say, why? Well, because the protists are eukaryotes that evolved from the prokaryotes. Well, plants and animals and fungi don't pop out of nowhere by magic. They have to come from something. Plants, animals, and fungi have as their ancestors the protists. And the protists have as their ancestors the prokaryotes, the bacteria, and the archaea prokaryotes. More on that later. So you guys know that things don't pop out of nowhere by magic. Animals have protestant origins. Plants have protestant origins. Fungi have protestant origins. That's why these protists have organisms that are plant-like, animal-like, and fungal-like. You'll have to understand this history, this genealogy, for various questions that you'll see in the future. plants and animals and fungi all go back in time to certain types of protists, have protistan ancestors. And these protists have as their ancestors prokaryotes. And then eventually I'll get into the story of the origin of life, and we'll discuss the life-like cells, what are called protobionts, that gave rise to the prokaryotes. We'll do that later. I'll stop for a moment for any questions about comments so far on the introduction of the protists.
So did you say so prokaryotes, eukaryotes evolved from prokaryotes, and then protists evolved from those eukaryotes, or?
Well, the first kinds of eukaryotes that evolved on the earth are protists.
Oh, okay.
Yeah. So for example, we'll look at some amoeba on Wednesday. We'll look at some paramecia on Wednesday. These are protists, and these are some of the earliest types of eukaryotic cells that come onto the earth. Does that make sense?
Yes, thank you.
Okay. Other questions about comments so far?
You said protists no longer have it's separated into a separate kingdom of their own. So what kingdom do they belong to then?
Well, there's 25 different kingdoms, and I'm not interested in learning their names. So Chase, you can do the research on that for me and learn all 25 different kingdoms if you want. I'm not going to learn them, and I'm not going to test you guys on them. You follow me? So I've ignored it. It's not something that I find at all interesting. So we have the kingdom Plantae, the kingdom Animalia, the kingdom Fungi, but we no longer have the kingdom Protista. It's got 25 different names.
How about you give us extra credit if you know all 25?
Huh? You guys can learn all 25 for your own interest. Extra credit is unfortunately not available in this course. But that's a good question there, Ashton.
At least I tried.
Yeah. And in fact, when we get to the chapter on the protists, I will be very careful to cut down on a lot of that terminology. We're not going to get crazy about it. So that'll be one area you want to be very careful with. You guys are not going to have to do all of that chapter on protists, and I will be greatly reducing some of the taxonomic terminology in that chapter. But that's for lecture exam 3. We'll worry about that later. All right, other questions before I move further?
You said there's 25 different kingdoms of protists, or it's 25 different kingdoms?
Of protists. There's 25, just of protists.
Wow.
There's 25 kingdoms. Yeah, we've learned that they are incredibly diverse. And they've been on the earth for, prokaryotes trace their ancestry back to probably 3.7 billion. The protists probably trace their ancestry back to 2.5 billion. So they've been on the earth for billions of years, at least 2.5 billion. So there's 25 different kingdoms of protists.
And they fall under the domain of eukaryotes?
Correct.
Okay.
Correct. Yeah, the domain is eukarya. The domain is eukarya. And everybody who's in the domain eukarya is composed of eukaryotic cells. Yep. All right. Other questions? So today's lab is about a group of protists called algae. And algae are plant-like protists that are found in marine and freshwater environments. Some are only found in freshwater environments, others in marine environments. And when I say a marine environment, I mean oceanic water. The algae are photosynthetic eukaryotic organisms that are unicellular and plant-like. Algae, photosynthetic eukaryotic organisms that are plant-like, unicellular, and can be found in the oceans and can be found in freshwater environments. We can trace the ancestry of algae back to 2 billion years ago. 2 billion years ago, we're confident that there are algae on this planet. They became very diversified about 600 million years ago. Algae underwent enormous diversification. That means simply that they evolved a variety of different types. And that occurred about 600 million years ago. If we had to pick out important aspects of algae, it is that since they are photosynthetic, right? So think back about photosynthesis. They're able to take sunlight and make their own food. These algae are important oxygen producers. So a byproduct of photosynthesis is oxygen, gas, O2. And so algae play an important role in being important producers of oxygen gas. It's a byproduct of their biochemical reaction called photosynthesis. And remember, photosynthesis, they're able to take the energy from the sun and make their own food. They don't have to eat other organisms. They are autotrophs, right? Go back to the terms autotrophs and heterotrophs. Autotrophs make their own food. When we look at the algae, we would describe it as being polyphyletic. Poly, P-O-L-Y, phyletic, P-H-Y-L-E-T-I-C, polyphyletic. All this means is that the algae don't form one complete phylogeny. If you are polyphyletic, it means you have a group of organisms that have all been grouped together, but they actually have many phylogenies, and they do not all share an immediate common ancestor. Now, this doesn't mean that they don't share a common ancestor at some point in time, but when we say algae is an informal general name that's polyphyletic, it just means there's a lot of diversity in the algae, and you don't just have one nice phylogenetic tree. You have a variety of different phylogenetic trees within this group. Some of these creatures are more closely related to each other than others are, and some of them really aren't that closely related. So being polyphyletic means a group of organisms that have been grouped together, but they do not share an immediate common ancestor. The other important aspect about algae, you know, we said they're photosynthetic and they produce oxygen gas as a byproduct of their need to make food for themselves, is that they are an important food source for the ecosystems in this world. The algae are a major source of food for a tremendous number of organisms in the various ecosystems on this planet. Questions about comments so far?
Is
Say philo. Sorry.
Go ahead, Washburn.
When you said that they are poly.
Polyphyletic. Phyletic.
Yes.
That is to distinguish them from monophyletic. If you're monophyletic, you have one genealogy tree, one phylogenetic tree, and you're going to be rather closely related. But if you're polyphyletic, this means we got a variety of genealogical trees. We got a variety of phylogenetic trees, and some organisms classified as algae or in the group of algae aren't that closely related.
Okay, thank you.
All right. Other questions before I go on?
yeah. Cyanobacteria, they used to be called blue-green algae. What make them distinct from the algae you're talking about?
Well, cyanobacteria should not be called blue-green algae. Cyanobacteria are bacteria. Cyanobacteria are photosynthetic prokaryotes. So I'm not saying that there aren't people that will call it that, but they should not be called blue-green algae. If they are, those people are incorrect. Algae are eukaryotic cells, and cyanobacteria are not eukaryotic cells. Does that make sense?
Yeah.
So, I'm not saying that there haven't been times where people made mistakes. It used to be, there was a time when we had certain types of algae classified as plants. But we'll get into this more when we get into plant evolution. But we've cleaned that up. But cyanobacteria, and I'm going to test this. Cyanobacteria are prokaryotes. They are photosynthetic prokaryotes, and they are not algae. I'm glad you brought that up because that's something that's going to be tested. I think lecture exam 3. All right. Other questions?
I do. So since algae are polyphyletic, and you said they were like a group of protists, wouldn't that also like relate to protists since they're also polyphyletic?
I'm not sure I understand the question. Algae are protists. Now, don't confuse the word protists with prokaryotes.
Okay.
You with me?
Yeah.
Cyanobacteria are prokaryotic cells, which are different from eukaryotic cells. So that's why cyanobacteria are not algae. Algae are made up of eukaryotic cells.
Oh, my question was not about cyanobacteria. I was just asking if we could call protists polyphyletic.
Yes, definitely.
Like
Protists would be their poly kingdom, right?
Okay.
Yeah, protists would also be described as polyphyletic, definitely. Yep. Definitely. All right, other questions before I go further?
I have a question. I think you might have mentioned it and I missed it, but when did algae approximately evolve?
We have the evidence right now puts algae at about 2 billion years ago.
Okay.
And then they look like they underwent nice diversification about 600 million years ago.
Thank you.
Yep. All right. Well, was there a question?
Yeah. So I was reading a chapter last night, and they were talking about single cell eukaryotes that was first appearing when prokaryotes started appearing on the Earth. So are there single cell single cell algae? because algae are under
Yeah,
eukaryote, so they
yeah,
would
yeah,
be.
yeah. Algae are single-celled eukaryotes. Yeah.
Oh, okay. So I
Um,
was under the impression that eukaryotes are all multicellular.
no,
Okay.
no, 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 no. Nope, not at all. You have with eukaryotes, you have multicellular forms, you have colonial forms, and you have most eukaryotes are unicellular. More on that in a little bit. But yes, most eukaryotic cells are single cells. And from the evidence we have right now, we have prokaryotes, not protists, but prokaryotes about 3.7 billion years ago. And then in around 2.5 billion, we start to see the first eukaryotes. And those would be the first types of protists. Okay. All right. We ready to move forward? All right. Here are the algae you guys have to do. Unfortunately, one of the reasons I really dislike how we're having to teach a class right now is I spent several hours yesterday just putting this stuff in eLearn. So, you know, I go to teach a lab, and what would normally take me 30 minutes to get ready to teach the lab takes me 3 hours of work to get the lab ready. I know, I'm whining. But here are the algae you guys have to do. And you'll have to, so for example, this first type of algae is from the genus Chlamydomonas. You'll have to know its phylum name. The phylum name is Chlorophyta. Its genus name is Chlamydomonas. And you can see that it's a single-celled unicellular type of green algae. What you guys are going to have to do for Chlamydomonas is you're going to have to be able to identify images as well as know any information I present about the Chlamydomonas in our little presentation here. So on your second lab exam, you might have images, and I'm not just going to take images from the ones I have here. You guys should go, well, you have your laboratory manual you can look at. I highly recommend you get the photo atlas. If you go back to the syllabus, you'll see the photo atlas. If you don't want to purchase the photo atlas, which I highly recommend, you can go to Google Images. So you guys will want to start to build a little file of images. You want to take the ones I have here, and you also want to have some other images. So I'm not promising you that on the lab exam, you'll necessarily have just the images that I have here. Let's start off with a diagrammatical image of Chlamydomonas first, like you can see here. This is from your textbook. You can see right here is a Chlamydomonas cell. They're unicellular. They're biflagellated. You can see the little flagellum right through there, little flagellum right through here. The little flagellum allows them to move, obviously. They are photosynthetic. You can see the chloroplast right there. And they have a little structure called the stigma. The stigma is like an eye spot, this little red structure right there. The stigma will appear as a reddish, light absorbing spot in the cell of Chlamydomonas. And the stigma allows for Chlamydomonas to track light. Well, tracking light is a good thing if you are photosynthetic. So Chlamydomonas are single-celled creatures, single-celled protists, or single-celled eukaryotes that spend time in the water. You can find them in the soil, wet soil. You can find them in lakes. You can find them in ditches. This is information you can get from your lab manual, by the way, where they sit around capturing sunlight and making their own food for themselves. You guys do not have to worry about asexual and sexual reproductive cycles of Chlamydomonas at this point. I'm just using this picture here so you can see what Chlamydomonas looks like. You do not have to worry. You will be doing a lot of life cycles as this course goes on, but you don't have to worry about the asexual and sexual life cycle of Chlamydomonas.
Would we need to know anything about like the super groups it talks about in like the photo atlas?
You'll find that I'm not interested in those terms. So you guys will need to know any terminology that comes out of my mouth. If I refer to supergroups, the answer is yes, you got to do them. If you don't hear them from me, you can learn them for your own interest level, but you're not going to be tested on them. Does that make sense?
Yes, thank you.
So the terminology you guys will have to do, you'll know what terminology you need to do by how I'm describing them. So phylum Chlorophyta, genus Chlamydomonas, and these are unicellular green algae. Now down here, notice there's a big difference between the diagrammatical view of Chlamydomonas and this image down here. The thing, you have to look around here. Here you can see a flagellum, but they're actually biflagellated. You guys can see my black pointer, right?
Nope.
So there you can see a flagellum, but they're biflagellated. Over here is probably a good one to be able to see the nucleus. So the black pointer's on the nucleus right there. Kind of looks like a little bulge or maybe a nickel inside there. That's the nucleus. And then it's probably hard for you guys to see, but right through here is the chloroplast. So let me show you up here. Right there's the chloroplast. That gray structure in the middle is the nucleus. In this image down here, the chloroplast is here, and that's the nucleus. The other thing I don't like about this cell is we can't see the flagella. But that's, you know, this is an actual image of Chlamydomonas, a group of them. So we can see a flagellum there, but it's just not one flagellum. They're biflagellated. So why are you learning about Chlamydomonas? Because we want to get you guys exposed to the diversity of life on the planet. And eventually, when we get into the more into the chapter on protists, we're going to find out that Chlamydomonas helps us explain the story of plant evolution. Plants don't pop out of nowhere by magic. They have protistan ancestors. And biologists know that plants have unicellular, plant-like algal ancestors that were eerily similar to whom? Chlamydomonas. We're not saying that plants evolved from Chlamydomonas, but they evolved from a Chlamydomonas-like algae. We know that plants have as their ancestry green algae. And so that's why we're doing Chlamydomonas. A Chlamydomonas-like unicellular protist billions of years ago in time gave rise to plants. So check out the diagrammatical images, check out the real image. I would say that you should also Google a couple of images and start to build a pictorial profile of Chlamydomonas. You can go to your lab manual and make sure that you have taken notes of what I've spoken here. So for example, do you think I might ask you what the stigma does on a lab test?
Flagella is to swim.
The stigma?
No, the stigma is to take the sunshine.
The stigma is an eye spot. Well, yes, you could say that, yes. The stigma is an eye spot that's reddish in color and absorbs light. And it allows for the Chlamydomonas to track sunlight, which it uses for photosynthesis. So you'd be surprised how many students, although I think you guys are starting to make some adjustments, they'll be going, lab test isn't for a while. I'm bored. I don't really care about Chlamydomonas. Meanwhile, they'll be affected on lab exam 2 when I ask questions about the stigma and any other comments here about Chlamydomonas. I know what I've said in class, and I'm the one that prepares the lab exam. So Chlamydomonas, here's an interesting thing. Chlamydomonas are haploid. They're not diploid. Haploid means they have one complete set of genes. Chlamydomonas, and you guys need to recognize that Chlamydomonas is the genus name, not the phylum name and not the species name. Chlamydomonas are haploid cells. All right, I've said all I need to say about Chlamydomonas. Questions before I go further? Oh, look here, guys, right there, stigma in bolded. I just gave you a test question. Shame on you if you miss it. I usually wait to discuss the chlorophylls in the lecture format, but you'll see that Chlamydomonas does have chlorophylls that are virtually identical to what is found in plants. All right. Questions about Chlamydomonas?
You said you're going to use pictures from the photographic atlas too for the
I can,
lab exam?
yes. Did you get the, yes. I can potentially use pictures from the photographic atlas, yes.
Yeah, I looked over it and I couldn't find the phylum kingdom for Chlorophyta.
Okay.
Yeah.
Yeah, I guess it probably depends on which photo atlas you might be using, but
I have
yeah,
the
the
8th
phylum.
edition.
Okay.
44.
The
So is this
phylum
one?
is Chlorophyta. Yeah, that's the one I want. Yeah, that's the one I want you to have. Yeah. You'll see the phylum name in your lab manual.
Chase, it's on page 44.
Oh, thank you.
You're welcome.
Oh, I see it now. Thank you.
And yeah, I will also take images from the internet. So if I take an image from the internet, the image will be similarly enough. It'll be similar enough to the images I've provided here or the ones that are in your photo atlas that you should be able to identify it. But that's
I
why
have
I'm
a
saying,
question.
yeah, hang on. But that's why I'm
Okay.
emphasizing you should also go to Google Images and get a couple look at Chlamydomonas as well. Morgan.
So if you like use images from Google, are you going to use ones that are like similar to the ones that are like you showed us right now? Or are you going to like use ones that are like that look different to like throw us off? I just don't want to like, I don't know. I don't want to be thrown off by something that is a different image compared to what we were shown. You know what I mean?
Yeah, well, Morgan, I have no desire in seeing people do as poorly as you guys did on my first test. That's not my goal. So I will be very careful to try to use images that are close enough to the images that are in your textbook. So this image here's from your textbook. This image here is from your lab manual. And then you're expected to purchase a photo atlas and the images from the photo atlas. It will be my judgment, but I will be very careful to pick images, and I never deliberately try to mislead students. You guys, my tests are tough enough that I can be straightforward with what's going to be on there, and students will still have some issues. Do you follow what I'm trying to say, Morgan?
Yeah.
I do not waste any time purposely trying to be tricky. That's not the intent of what I do. So I can promise you that the images I choose are a close approximation of what you've been studying if you study your textbook image, your lab manual, and your photo atlas. But that's
Okay.
why I'm also suggesting you should probably take a look at some of the Google images, right?
Okay. Thank you.
Yep. But I can promise you that they are going to be very similar. The trouble is for students who try to cram this, and you're not going to be this way. But the trouble is when students try to cram all this in 2 days before a lab test, and all they've done is memorized one image over and over and over again, if they get an image that is slightly different, it throws them off. You follow me?
Yeah.
So the way to not be thrown off is to work on this a little bit at a time, many weeks in advance, and to look at a variety of different images. Okay?
Thank you.
Yep. All right. Hang on just a second. Okay. Other questions about Chlamydomonas? Okay, good. Sorry. Some of you guys know that I have a tendency to respond quickly to emails because my phone lights up whenever students send me something. And if I'm near my phone, boom, I get it. And that's why I'm so quick. And I just got an email from an evolution student that said they were having trouble with the videos. And so I started to panic. But then they said, never mind, it's been solved. So that is good. All right, that's good news. Sorry. Let's move on. Our next example of algae is from the genus Volvox. The same phylum name, phylum Chlorophyta, genus Volvox. This is also a type of green algae, freshwater, and we're going to call it multicellular, but we're going to see here that there's been a lot of debate about this. Some have called it colonial in the past, more on that in a little bit. So same phylum as Chlamydomonas, but a different genus. Genus name is Volvox. Let's take a look at it. Now, let me see where I want to start. I think I'll start here. This is an image from your textbook. And if you look up here, well, here's Volvox. Volvox is this ball. Look at this ball right through here. And then it's got these little green balls inside of it. Volvox, imagine, you know, a plastic kickball. I got to stop looking at that. I'm getting distracted by my phone. You know what you do when you're distracted by your phone? Turn it off or turn it over. I got students sending me stuff here. I got to ignore those guys. So imagine you have a plastic kickball, right? And so this round structure is the kickball. And inside of it are these little green balls here. Notice the surface of the ball has these little tiny dots. You see these little dots right here? Not these big green dots, these small little dots here. Do you guys see those? Those small dots are these cells up here. In this image, what they've done is they've magnified the wall. So here's the wall of Volvox. And they've magnified this wall here through here, and the wall would be just like the surface through here. And that's what they're showing you here. These cells through here are eerily similar to Chlamydomonas. They would be best described as Chlamydomonas-like cells. And so these little cells through here, and you can see the background of them, that's what these little dots are right here, where my black pointer is. All those little dots right there are these Chlamydomonas-like cells. You can see the flagella. They're actually biflagellated. You can almost see two coming off through there. They are photosynthetic. These, you know, so Volvox is not described as unicellular. Volvox used to be described as colonial. They've switched it lately, and they're saying that it's a multicellular algae. And so one of the things I want to do is differentiate the term colonial from multicellular. So that's what I want to work on for a moment. What's the difference between the term colonial? You can see I scratched it out here. And the term multicellular. Well, what's the similarities? Well, both terms, colonial and multicellular, refer to groups of cells. Both are multiple sets of cells, right? So Volvox is not unicellular. Volvox is this whole entity here. Here's another Volvox group over here, and another Volvox over here. It's actually a very pretty organism. You know, the way they float around in the fresh water, if you see them on video, it's a beautiful thing as they rotate. The Volvox is able to move because of the Chlamydomonas-like cells that have those flagella. So the beating of the flagella helps the Volvox rotate. and move. Well, the similarity between colonial and multicellular is we're referring to a group of cells. So what's the difference? The difference is the degree of specialization. In a colonial arrangement, you have a group of similar cells that have a low degree of specialization. While in a multicellular organism, you have a group of similar cells that have a high degree of specialization. And you might say, well, what do you mean by a high degree of specialization? Well, in multicellular organisms, you have specialized cells doing specialized functions. While in a colonial arrangement, you have a much lower degree of specialization. We would say in a colonial arrangement, you have a much higher degree of independence. While in a multicellular arrangement, you have a high degree of interdependence. You guys see the difference between interdependence and independence? Our children, as we raise them, were very much strongly interdependent upon their parents. As they head off to college, they start to transition from interdependence to independence. So we're going to make the adjustment. Please understand that this picture was taken from an edition that back in the time when they considered Volvox to be colonial. But they have changed that now, and they've made the determination that Volvox is multicellular. But the fact that there's this argument about whether they're colonial or multicellular just goes to show you that nature is not concerned about us humans trying to categorize this stuff. If you want to call Volvox multicellular, which that's how we will describe them because that's the new edition's version, you have to understand, guys, that whether you're multicellular or colonial is not an on-off switch. It's a continuum. And so think of a continual distribution. On the left-hand side, you have fully colonial organisms, which have a very low degree of specialization, such that if you were to separate that group of cells, they could live on their own. That's how little specialization they have. On the other side of the continuum of distribution, you have a multicellular group that is very highly interdependent. And if you take out some of the cells from that, the whole organism dies. Think about your body. Can you guys live without a heart? And I don't mean to say start thinking about being in a hospital on a machine, right? But if we take your heart out, are you going to live? Obviously not. Complex vertebrates are highly specialized multicellular organisms. Well, Volvox is a very simple form of multicellular. Or you could say it's a high-level form of colonial. You guys with me? We got a continuum of specialization here. So that's why scientists have been struggling with this. Volvox is the perfect intermediate living form that is on the verge of the transition between what we might call a colonial organism and what we might call multicellular organism. nature doesn't make it easy for us to categorize things because nature is an evolved world and it's not interested in humans trying to understand it. And so that's why scientists have gone back and forth about whether Volvox should be colonial or whether it should be multicellular.
So can I assume that they all have the same characteristics, or they have different characteristics that if they are in between?
We will, we're going to say that Volvox is multicellular because that's, you know, people have decided that that's the best way to describe it. But I want you to understand that they're right on that cusp. In our definitions of colonial and multicellular, they're right on that boundary of those definitions. Does that make sense? So
Yes.
Volvox is multicellular, but it's multicellular on kind of a lower end of the multicellular spectrum as far as the degree of specialization. So this is an image here of Volvox. So I will admit, guys, let me be careful. And your photo atlas may use the term colony. I may at times call these Volvox colonies, meaning a group of cells. But technically, we shouldn't call them colonies because they're no longer considered colonial. Those little green balls inside are their babies, often referred to as daughter colonies. In fact, I may even use that term on your lab test. If I say daughter colonies, try not to think that I'm trying to be tricky. You guys with me? Technically, we shouldn't be using term colonies because they're multicellular. But if I put a black pointer on a lab test on one of these green dots in here and ask you, is it called a daughter colony? You guys should say what?
Yes.
Yes. You guys understand?
What
So
did you say again?
I said, if I put this black, if I see my black pointer here,
Yes.
it is on a daughter colony of Volvox. These are the babies. These little green balls inside were the asexually reproduced babies of the Volvox. Eventually, they will leave the Volvox ball here. So the large spherical structure is the Volvox multicellular protist. The little green balls inside are the babies. And if we call them daughter colonies, don't say, oh, well, this isn't a colonial form, so they shouldn't be called daughter colonies. It's kind of leftover terminology of when they were used to be considered colonial.
What?
Technically, we should probably call them the daughter, I don't know, the daughter little babies, whatever. But these are called daughter colonies. You guys follow me? Well, the Volvox didn't appear out of nowhere. Volvox has its ancestors, which are very similar to Chlamydomonas. Volvox, if Volvox has eerily similar Chlamydomonas-like cells, this would suggest that they are related. So the ancestors of Volvox would have been groups of cells that would have been like Chlamydomonas, but would have been more of a colonial form before multicellular evolved. So why are we bringing this stuff up? Well, we're trying to give you guys a sense of how things evolve. You begin with unicellular organisms. In time, unicellular organisms will form groups of cellular arrangements that are colonial in nature. And when I say colonial in nature, I mean they have a low degree of specialization. Most cells are pretty much independent of each other. If you separate those cells in a colony, a colonial form, they're probably going to live because they still have a lot of independence. But then over time, a colonial form can undergo evolutionary change and become multicellular. If we describe something as multicellular, we have a group of similar cells that have a higher degree of interdependence, meaning there's a high degree of specialization where cells are relying upon other cells to do specialized jobs. That's what we called the term differentiation, right? That whole discussion about developmental biology dealt with how cells differentiate. Well, in a multicellular organism, you get a lot more differentiation. Cells specializing to do specialized functions. All right, so here's another image of Volvox. Again, the black pointer is on what we'll call the daughter colonies. The black pointer now is on a Chlamydomonas-like cell. If I put the black pointer out through here, you know, this whole thing, the whole thing is Volvox. Here's another Volvox group. The whole structure is Volvox. The little tiny dots there are the Chlamydomonas-like cells. The bigger dots are the Volvox babies, the daughter colonies, produced by mitosis. If you guys see the term like oogamy down here and I haven't explained it, don't worry about it. Trust me, I'm going to cover the language that you guys need to understand. Here's a Volvox group. Now, if you guys don't have to worry about the zygotes and the egg and vegetative cells, here's one that's specializing in the sense of being able to do sexual reproduction, and you don't have to worry about identifying zygotes, vegetative cells, or eggs. All you'd have to know with this image is that you're looking at a Volvox, and technically, since all you guys have to do is the daughter colonies, we would call what we would call two is on the Chlamydomonas-like cells, and one, for our purposes, is on what? What are we calling one?
Daughter colonies.
Daughter colonies, right. We're not going to worry about the fact the Volvox can reproduce asexually or sexually. You guys have to worry about its asexual reproduction, which means these daughter colonies down here were produced by mitosis.
So the 3 is what? Is still the one daughter colony? One daughter?
Over here?
The 3, yeah?
Yeah, I wouldn't test 3, but yes, for your purposes, 3 is a daughter colony. We're not going to worry about eggs and vegetative cells and zygotes here. All right, so Volvox, a multicellular green algae, right on the borderline of colonialism. Phylum Chlorophyta, genus Volvox, has biflagellated Chlamydomonas-like cells. You should probably try to YouTube a video of it and watch them spinning in the water. Beautiful organism. All right, questions about Volvox?
But I don't see the biflagella there.
Right, because it's not magnified enough. Each one of these cells here is biflagellated. That's why I like this image down here. This has magnified the wall of Volvox enough where you can see the little flagella. But you're right, don't be confused. This thing right there has two flagella, you're just not seeing them. Okay,
Thank you.
other questions about Volvox?
So that cell there, that cell there is one is able to do sexual reproduction, and some cells or
Well, all Volvox have the ability to do asexual versus sexual.
Okay.
You guys have to worry about the asexual form.
Okay. So
All
the
right.
daughter colonies are also known as zygotes, and the
Well, technically not. No. The
Oh.
daughter colonies are made by mitosis. Zygotes would be formed by sexual reproduction. So I picked this image because it's similar enough to these down here. But you guys don't have to worry about zygotes because it's very difficult for us to differentiate the zygote from the daughter colony, right? There's a way to do it, but I'm not interested in teaching you guys how to do it. You follow me? So
yeah, I
we're
was
just
curious.
we're just going to pretend that that zygote there is a daughter colony because it looks close enough to a daughter colony down here. But there is a way to differentiate them. It's just I'm not interested in getting into that. As you'll learn, the more you learn about something, the more there is to learn about that something.
That's true.
All right. Other questions about Volvox?
So I'm assuming the vegetative cells are different from the
No, it's just a funny name. I don't like the term vegetative. You'll never hear me use that term. The vegetative cells are the Chlamydomonas-like cells.
Okay.
I really don't like, you'll never hear me say vegetative. Every time I see that term, I just think of vegetables. I'm more like you guys than you think.
So when I go fishing and I see this green stuff, they are not Spirogyra or some.
No. Next one we're doing is Spirogyra. My guess is when you go fishing and you see this green, stringy, slimy stuff, that is Spirogyra. That's our next one. The Volvox colonies would probably be real hard to see. They're so small. I don't think you'll need a microscope to see them. While the Spirogyra is something that you can see without the microscope.
Okay.
Other questions about Volvox? All right. All right, so Washburg has just mentioned Spirogyra. That's our next one. Spirogyra is also from the phylum Chlorophyta. So the fact that all of these are in the same phylum means that they are reasonably closely related. The genus name is Spirogyra. Let me modify that. I don't need a comma there. I just did this yesterday. Phylum Chlorophyta, genus Spirogyra. Common name water silk. Looks like green silk in the water. This is a type of freshwater green algae, multicellular as well. We might argue, though, that the degree of multicellularity here for the Spirogyra is probably a little higher than that of the Volvox, because there's not been much argument about Spirogyra's degree of specialization. So it probably has a higher level of interdependence of the cells than Volvox does. All right, here's an image from your lab manual. Here's an image from the photo atlas I wanted you guys to get. All right, let's start with this one right here. Here is the Spirogyra is described as a filamentous green algae. You can see it right here, filamentous green algae, because the cells are connected one after another in a linear arrangement. So over right here is a filamentous strand of Spirogyra. You can see here's where the two cells join. There's where the two cell walls meet right here. So right through there's one cell, there's another. You can see that's the demarcation line between two cells here and here and here. What do you think that little green dot in the middle is?
Nucleus?
That's the nucleus. Yep. Yep. It's called Spirogyra, which sounds like a rock and roll band, right? Because of how the chloroplasts spiral up inside them. So the black pointer's on the nucleus, but then if you look inside there, you get this spiraling effect of the chloroplast. You can actually see this nicely down here. Let me move you guys here. Right through here, you can see that. So here's the dimensions of the cell go here. Here's the cell wall. Here's the cell wall. There's the cell wall. Here you can see the nice spiraling nature of the chloroplast inside of it.
Are the filaments just like composed of the chloroplast?
The filaments is just the cells connected to each other. So this is a piece of one filament. So a filament is like a string of cells.
Okay.
You got this cell here connected to that one, connected to that one, it goes on and on and on. So it sounds like Washburg is familiar with this stuff. It looks like little green strands of strings, maybe green silk, right, in the water, right, Washburg?
I go fishing, so
You go fishing. Well, sometimes if the fish aren't biting, that's all you're gonna get, right?
Yes.
Get some Spirogyra.
So that's probably what's growing on our pond.
Oh, it's very common in ponds. Yeah, if you go out there and look for green stringy stuff, very high chance that that's Spirogyra. Yep.
Okay.
So you can see right here, Spirogyra cells next to each other forming a filament. And what's interesting is what is next to them. This is Spirogyra in the mating game. So this is one Spirogyra filament. This is another Spirogyra filament, and they are going to mate. This is their form of mating here. This, the way they go about mating is referred to as conjugation. That's why these little tubes that are forming between these strands are referred to as conjugation tubes. This is their type of sexual reproduction. So one Spirogyra filament is come up to side by side with another Spirogyra filament, and it has formed a, they've grown these little tubes toward each other and have connected. They're called conjugation tubes. What you can see next to this is that one of the filaments is now sending its DNA over to the other side. You guys don't have to worry about plus gametes and minus gametes, but essentially what's happening is this cell is sending its DNA over to the other side. And what you see in this image over here, what look to be like little eggs, those are the babies. Those are the diploid zygotes. So this is the act of conjugation or the act of sex for these algae. They've come up next to each other, formed these interconnecting tubes called conjugation tubes. And then one strand is going to send its DNA on to the other strand. These little things. Now, so notice that these, do you think the filament here with the black pointer, do you think these cells are going to survive or die? They're going to die. But they've sent their genetic material, which originally was haploid. So a, well, if we want to call it an adult form or a fully differentiated, mature, filamentous form of Spirogyra, those individual cells are haploid. But now they're undergoing sexual reproduction and combining their genes. So haploid plus haploid means what?
Diploid.
Diploid. As you guys know, diploid means two complete sets of genes.
So does the one chloroplast only transfer its genetic material when it's like in the adult stage or when it's about to die?
Well, it's not the chloroplast that sends its genetic material. It's the nucleus. So back to this cell, do you see that little green dot right there?
Mm-hmm.
That has one complete set of genes. So it's the nucleus that's going to send its DNA over.
Okay, and does it do that when it's like about to die, or can it do it at any stage of development?
I'll be honest with you. I don't know for sure, but I don't think it does it because it's about to die.
Okay.
It's just a part of the process of getting an opportunity to mate. And yes, these cells will die here, but it lives on in its offspring.
Okay.
So what you see right through here are the zygotes, the little babies. And then if you wonder, well, how do you get back to this filament? What will happen is each one of these zygotes will disconnect from the filament. Look at this last sentence down here. I don't know, can I highlight that? Maybe I can't highlight that last sentence down there. The zygotes will then undergo meiosis, producing the haploid cells that will then undergo mitosis to make new filaments. So if you're wondering, how do you go from a zygote to a filament over here? The zygote will undergo meiosis, making haploid cells that are genetically different. And then those individual haploid cells will grow their own filaments by mitosis. So let's take this little Easter egg right here, the zygote. It will undergo meiosis, forming ultimately 4 genetically distinct cells that are all haploid. And then each one of those cells will form a new filament by mitosis. So because the filament is formed by mitosis, every one of these cells over here in this filament has the same DNA. It's kind of intriguing to consider that something as simple as this algae is going about doing similar life cycle things as animals do. The meiosis and the mitosis cell division process is virtually identical to what goes on with mitosis and meiosis in animals. Down here, you can see the spiraling chloroplasts. Here, you can see a filament through there. If I put the pointer on here, you probably should call it cell wall. If I'm there, it's the nucleus. If I'm here, what is it? What's this here?
Chloroplast?
The chloroplast. Here, they've shown you the dimensions of one cell.
Where did you say the cell wall was?
Over here, you got the cell wall all up and down here, across there, down through here, and along through there. Over in this image, it's really easy to see it. You got cell wall here, there, there, and there.
Looks kind of like a deep double helix in DNA.
Yeah. Yeah, it does, doesn't it? All right, let me see my notes on this.
Is the cell wall that is called the chloroplast?
No, the cell wall is very different from the chloroplast. The cell wall, the pointer's on the cell wall here. The chloroplast is right there. A chloroplast is a tiny little organelle needed for photosynthesis. The cell wall is basically like a wall of your house in a way. So for me to go, no, it's different. It would be like you trying to tell me that the wall of my house is the same thing as my refrigerator. You follow me?
Yeah.
Very different. Chloroplast is very different in its structure and function. Cell wall is the enclosure or the boundary of the cell.
Thank you.
Yeah, let me look at my notes. So Spirogyra, a filamentous green algae found in freshwater ponds and streams. Phylum Chlorophyta. It's multicellular. It's obviously a plant-like protist, but it's not a plant. And it gets its genus name from the spiral-shaped chloroplasts. In fact, most of the stuff, if you guys ever see what you think is a plant in the water, there's an extremely high probability it's algae. There are very few aquatic plants. And those aquatic plants that do exist actually evolve from terrestrial plants. So anytime you look in the oceans or the lakes and streams and you think you're looking at a plant, there's an extremely high probability you're looking at what's called algae. There is a difference between algae and plants, and we'll be talking more about that difference when we get into the story of plant evolution. All right. Again, you'll want to look at other images of Spirogyra. Questions about Spirogyra?
I have a question.
Yep.
So would conjugation be considered different than mitosis since they're not copying the DNA?
Correct. Conjugation is their form of sexual reproduction.
Okay.
Yep. Conjugation is their form of sex.
But it's not considered sexual reproduction, is it?
Yeah, we consider it sexual reproduction because
Okay.
they form these little conjugation tubes and they send a set of DNA from one side over to the other. So they're combining DNA. Sexual reproduction just means you have two parents, right? So this zygote, this baby, has not come from one filament, but both of these filaments.
Hmm. Hmm.
So it's sexual reproduction. The thing you're probably thinking of, Chase, is you're probably confusing this, which is easily confused. I wish we didn't use this term. Conjugation with bacteria.
That's what I was thinking.
Yeah, in bacteria conjugate, prokaryotes conjugate, but it's a situation where one bacterium sends plasmids over to the other one. They're not both exchanging DNA, and it's not sexual reproduction. So I wish we'd use a different term, but the scientists that came up with these terms at the time, they weren't thinking about students having to learn this stuff eventually.
Thank you for
Conjugation
explaining.
here. What's that?
I think that's what confused me because I learned about bacteria.
Yeah, conjugation here is different than conjugation with bacteria, and I apologize, but I can't change their terminology. Bacteria, when they conjugate, one bacterium sends some of its plasmids over to the other one. But that's not sexual reproduction. They're not making a new baby from that union. But this form of conjugation here with these eukaryotes is a form of sexual reproduction. Sometimes people will call conjugation with bacteria, they'll call it bacterial sex, but they'll put parentheses around the sex. It's not really bacterial sex. It's not sexual reproduction. So part of that confusion is, one, we're using the same term, and two, I think people are a little misleading when they talk about what the bacteria are doing. You follow me?
Yes.
So conjugation is it just bacteria, or does that apply to the difference between how conjugation is used in prokaryotes versus eukaryotes?
Yeah, unfortunately, you have to realize that the two terms are differently applied here in eukaryotes and prokaryotes.
Okay.
Unfortunately. And I do apologize for that. But this type of conjugation here is legitimately called sexual reproduction. While if somebody's referring to conjugation in bacteria as sexual reproduction, that is not legitimate at all. In fact, it shouldn't be described as sexual reproduction. Because they're not, when the bacteria do it, they're not making an offspring. It's just that, you know, if you guys want me for the moment, just use this picture. If these were bacteria, because bacteria form these little tubes. If this were bacteria, one would come up next to the other one. They wouldn't be in a filamentous arrangement, but one would come up to the other one, and one would just give it a few plasmids. It doesn't give it all its DNA. It just gives it a few plasmids. And the other one doesn't give the other one anything. Conjugation with bacteria is one way. exchange. So, and they're not making a baby. You follow me, Chase?
Oh yeah, I get
So yeah, conjugation in prokaryotes is really very different than the way it's happening here with the eukaryotes.
Yeah, that's really weird how they didn't just find a different term for that. That seems like a very,
To me, we need different terms.
yeah.
I agree. But we got to deal with it. All right. Ignore this picture. This part of this picture just came in here because it was with this picture. We're not, you guys don't have to, we're doing Spirogyra, and this is not Spirogyra, so you don't have to worry about that image there. All right. We done with Spirogyra. All righty. I'm going to do one more, guys. I'm starting to fade. Zoom is not my favorite thing. On Wednesday, we have, it's a little easier to do that. So I'm thinking I'm going to do one more of these, and then we'll call it a day and get back to work on Wednesday and finish the dinoflagellates and euglena. And then we have to do protozoa. We got 4 protozoans to do. So let's do diatoms, see how I feel. I'm starting to fade. If we were in class together, I'd be energized. I'm like the energized bunny. I'm just fired up. The adrenaline's going. It's like showtime for me. Literally being on Zoom is kind of like being at a practice, while being in class is like being in a game. Game time is fun. Zoom time is practice. Not so much fun. It would help if I had more pictures of people though, but I understand that you guys are lucky I don't require you. Some faculty require you to put your, hey, I can see Kenny there. How's it going, Kenny?
I'm good.
So I see some images here. It's helpful to see your pictures. But mostly I see a black thing that says your name. I got a black square that says your name. That's what I'm seeing for most of you guys. I'm not complaining. Well, I am complaining, but I'm not going to force you guys to put your images up there. Hello, who was that? I saw somebody there. I don't see your name. Who is this?
Carrie?
Carrie. Hi, Carrie. Sorry. I need to start learning you guys' faces. I should have you guys have to introduce yourselves here. Hi, Carrie. All right. Let's go to diatoms. Notice diatoms. There's no taxonomic terminology you have to do. There's no phylum names here, no genus names, in fact. And you might say, well, why? Because there's such a diversity of diatoms. There's so many different phyla and so many different genera that we just call them diatoms. So you guys are going to like that. So no taxonomic terminology, guys. You don't have to do any phylum names, no kingdom names, no genus, species names. They're just called diatoms. You might say, well, what are diatoms? I've given a simple definition here. These are unicellular algae. You could also call them unicellular protists that are very abundant in the oceans and freshwater environments. And diatoms are intriguing as far as their structural arrangement and their beauty and shapes. Look at this. This is taken from your textbook. Diatoms have a diversity of shapes. Diatoms look like little glass cells in a way. If you look at these images, they look like they're little tiny decorative jewelry of some sort, and they look like little pieces of glass. Well, there's a reason for that. The cell wall of diatoms is composed of the same compound we use to make glass. So the cell walls of these diatoms is composed of silicon, or what you could call silicon dioxide. Silicon, S-I-L-I-C-O-N. Or you could call it silicon, S-I-L-I-C-O-N, or you could call it silicon dioxide. Silicon or silicon dioxide is what we use to make glass. The same mineral, the same chemical substance is found in the cell walls of these diatoms. That's why they look like little glass houses or little jewelry, little glass cells. Remember, these are unicellular algae, so they are photosynthetic. These things are extremely numerous in aquatic environments, in the oceans, for example. In fact, they're so numerous that they are a very important food source for a number of aquatic animals. Sometimes I jokingly say diatoms are kind of like the popcorn or the M&Ms or the peanuts. Pick your favorite little treat to snack on.
Those are different species.
Yeah, in this picture right here, you're probably looking at, well, those two look similar, right? But the difference between that one and over here is probably different species, may even be different genera. So that's why we're not going to worry about taxonomic names, because in this one picture, you probably got all kinds of different genera, if not some different phyla maybe. So they just call them diatoms. But a lot of aquatic animals like to feed off these things. They're like the popcorn of the ocean, or the M&Ms, peanuts of the ocean. They are unicellular algae. They're photosynthetic. So they're making their own food. They're capturing sunlight, making their own carbohydrate by way of photosynthesis. And then other organisms come along and eat them. Excuse me. You guys will want to remember the chemical compound that makes up their cell wall. You could call it silicon, or you could call it silicon dioxide, same substance we use to make glass. And these diatoms are referred to as the primary producers of the oceans. They are, as primary producers, what we mean by that is, since they carry out photosynthesis, they play a very important role in beginning the energy transfer that occurs in aquatic ecosystems. There are a lot of things that like to feed on these diatoms. If anytime you hear something as a primary producer, that means you're an autotroph. You're photosynthetic. All right, let's see. What else do I have here? Oh, I like this. This is another image from your textbook. Look at the definition. Diatoms, unicellular algae that have this unique glass-like wall made of silicon dioxide. And I like this image because you can see the really intriguing architecture of the cell. Beautiful shapes, almost like jewelry. And here's another image here of a variety of different diatoms, a variety of different shapes, all carrying out photosynthesis. So the one thing you have to be careful with diatoms is make sure you look at enough pictures. There's a good picture, great picture, but you'll need to do some Google images too. And look at enough pictures of diatoms. You guys with me? So diatoms, yep, got that. Type of algae, I said that, okay. Questions about diatoms? Questions about diatoms? Okay. I'm starting to fade here a little bit. Here, one thing I do want you guys to note is eventually, let's see. Can you guys see anything yet?
On your screen or on our side?
My screen.
We can see what you see.
Okay. Let me make lab exam 2 review slides. Let me make it visible to you guys. Visible. I want it visible. All right, visible. Okay, so now that's visible. I want this out of my way. I got, oh, lost you guys. Move that over here. Huh. I'll get you guys back here. All right, here we go. All right, so now in the table of contents, can you see where it says lab exam 2 review slides?
Yep.
So you click on that, you come over here. Eventually, when we get through all this material, you guys will want to make sure that you go through this PowerPoint presentation and work on these slides. I don't plan to go through this specifically with you. You guys will have the knowledge if you pay attention in the lab sessions that we do. You will have the knowledge to be able to figure these out. But I want to go to see if I find anything like we did today. Let's see here. Aha. What is that thing? Did we do this today?
Yes.
Yes. That is Chlamydomonas. So where's Morgan? Morgan, does that look like Chlamydomonas?
Yeah.
Yeah. So you got the eye spot you got to learn about. You got the chloroplast there. You got the nucleus. You got the biflagellated condition. Let's see here. You guys will be learning how to identify all of these eventually. What's that look like?
Tom's.
Diatoms.
Diatoms. Yep. These are the dinoflagellates. We haven't done those yet. Ah, what's that look like?
Chlamydomonas.
Chlamydomonas, yes. There's the biflagellated condition. There's the cell. You got a group of Chlamydomonas right there. What's that there? That's the same image I showed you guys. You guys with me?
Chlamydomonas.
If I put the black pointer on that, what is that? The black pointer's on what right now?
The nucleus.
The nucleus. Thank you very much. Oh, look at these pretty things. What is the black arrow on right now?
The daughter colony.
colonies.
So
Yep. If I put the black arrow, my black arrow's on what right now?
baby cell.
Vegetative cells.
Chlamydomonas-like cell, right? While these black arrows are on daughter colonies of Volvox, right? Ah, what are those?
Tom's.
The diatoms.
Diatoms. Those are something else we'll discuss some other time. There's 70 slides there.
Wow.
So my point was I wanted to show you that the stuff we learned today will help you identify some of these practice. But this is, the point of this is going to simulate your lab exam 2. So you would get an image like this. You click on, you get an image like this, and then I will have some questions about it. Does that make sense? My point to you is, let's see, we've lost a few members. We are up to 29 students. Some have decided they drop out here today early. But my point is, I will just tell you guys, it will be helpful for you to spend some time working through a PowerPoint presentation that I spent time in my life making that will help to get you prepared for the lab exam 2. Do you guys follow me? All right. Now, I'll be honest with you. I'm burned out here. So Wednesday, I have 4 things the protozoans do. I still got dinoflagellates. We won't have as long of an introduction. I think we can finish things on Wednesday. Unless you guys want me to do dinoflagellates. What's the vote? Have we had enough?
Yeah, you can do it on Wednesday.
Okay.
Yeah, do it on Wednesday.
All right, thanks. That's what I wanted to hear. So we will start with dinoflagellates, and then we'll go to euglena, and then we'll do the protozoans. We'll finish that on Wednesday. You know, I don't want you guys to think that I'm cutting class. Why am I flexible about this? Well, you guys know why. I've spent my weekends sometimes making these videos for you. So because I'm making these videos is why I don't have to meet 8 hours a week on Zoom. My chairman approved this way of doing this. In fact, technically, I'm only required to meet like 4 hours a week on Zoom, but we usually meet more. But if you're
We are
wondering,
now on hybrid though.
what's that?
I say we are now on hybrid though.
Yeah. But if you're wondering why I'm flexible with Zoom, it's because of the videos and